all of you students. Here, the new topic is mineral deficiencies disrupt plant metabolism and function. किस तरह से मिनरल की डिफिशियंसी होने पे प्लांट का कौन सा मेटाबॉलिज्म जो है वो डिस्टर्ब होता है या प्रॉपरली फंक्शन नहीं करता। इस सेक्शन में हम इस बुक की हेल्प से यही सीखेंगे। सो हाईलाइटेड किया हुआ है जो इम्पोर्टेंट पॉइंट है और रेस्ट ऑल जो है वो डिस्क्राइब करेंगे। इन हाइड्रोपोनिक कल्चर withholding of an essential element can be readily correlated with a given set of symptoms for acute deficiencies. तो तरह तरह से हम किस तरह से हम चेक करते हैं कि कौन से एलिमेंट से प्रॉब्लम आता है, तो वो हम देखते हैं जब उस एलिमेंट को हम उस जगह से हटा देते हैं और तब देखते हैं कि उसकी वजह से कौन सा प्रॉब्लम आ रहा है। ठीक है। Both chronic and acute deficiencies of several elements may occur simultaneously. Deficiencies of excessive amount of one element may induce deficiencies or excessive accumulation of other. Some virus-induced plant diseases may produce symptoms similar to those of nutrient deficiencies. Nutrient deficiency symptoms in plant are the expression of metabolic disorders resulting from the insufficient supply of an essential element. Nutrient deficiency symptoms in plants are the expression of metabolic disorders resulting from the insufficient supply of an essential element. In general, the essential element function in plant structure, metabolic function and osmoregulation of plant cell. Most specific role may related to the availability of divalent cations such as calcium or magnesium to modify the permeability of plant membranes. In addition, research continues to reveal a specific role in these elements in plant metabolism, like calcium acts as a signal to regulate key enzymes in the cytosol. Most essential elements have multiple roles in plant metabolism. So यहाँ पे हम देखेंगे कि deficiencies में किस तरह की deficiencies होती है. When relating acute deficiency symptom of particular element, an important clue is the extent to which an element can be recycled from older to younger leaves. यहाँ पे कुछ एलिमेंट्स ऐसे होते हैं प्लांट्स के केस में जो मोबाइल होते हैं, यानी ओल्डर लीफ टू न्यू यंगर लीव्स में वो मूव कर जाते हैं। कुछ ऐसे एलिमेंट्स होते हैं जो इमोबाइल होते हैं, जो बिल्कुल मूव नहीं करेंगे। सम एलिमेंट्स लाइक नाइट्रोजन, फॉस्फोरस, पोटेशियम मूव फ्रॉम लीफ टू लीफ and others like boron, iron and calcium are relatively immobile in such plant species. If an essential element is mobile, deficiency symptom tend to appear first in older leaves, क्योंकि वहाँ से वो एलिमेंट जो है मूव कर गया यंगर लीव्स की तरफ। सो वहाँ पर वो ओल्डर लीव्स में प्रॉब्लम क्रिएट करेंगे। Deficiency of an immobile essential element will become evident first in younger leaves. तो यहाँ पे हम देख सकते हैं कि ये मिनरल एलिमेंट्स का चार्ट है जहाँ पे दिखाया गया है कि किस तरह से कौन कौन एलिमेंट मोबाइल है और कौन कौन इमोबाइल है। राइट, वी हैव ऑलरेडी डिस्कस्ड in previous lecture that how these elements are categorized, so the deficiency will also be discussed according to their groups. So group 1, deficiencies in mineral elements, nutrients that are part of carbon compound. Carbon compound is very important source for plant life, because everything made up of organic substance, and organic is the carbon. So the first group consists of nitrogen and sulfur. Nitrogen availability in soil limits plant productivity in most natural and agricultural ecosystem. Nitrogen is the mineral element that plant requires in greatest amount. It serves as a constituent of many plant cell components, including amino acids and nucleic acids. Thus, nitrogen deficiency rapidly inhibits plant growth. In such a deficiency persists, most species shows chlorosis, that is yellowing of leaves. Under severe nitrogen deficiency, these leaves become completely yellow. Younger leaves may not show these symptoms initially because nitrogen can be mobilized from older leaves. Nitrogen deficient plants may have light green upper leaves and yellow or tan lower leaves. When nitrogen deficiency develops slowly, plants may have markedly slender and often woody stem. This woodiness may be due to the buildup of excess carbohydrates that cannot be used in the synthesis of amino acids. Carbohydrate not used in nitrogen metabolism may also be used in anthocyanin synthesis, leading to accumulation of that pigment. This condition is revealed as a purple coloration in leaves, petioles and stem of some nitrogen deficient plants like tomato and certain varieties of corn. So the next one is sulfur. Sulfur is found only in two amino acids, that is methionine and cysteine. So it is the constituent of several coenzymes and vitamins also. The symptom of sulfur deficiency is related to few times similar to nitrogen deficiency like chlorosis, stunting of growth and anthocyanin accumulation. The chlorosis caused by sulfur deficiency arises initially in mature and younger leaves rather than in old leaves because sulfur cannot be mobilized from older to younger leaves. Now we will move towards the next, that is group 2, deficiencies in mineral elements that are important in energy storage and structure integrity. This group consists of phosphorus, silicon and boron. Right, so we start with phosphorus. Phosphorus, found as a phosphate PO4 3- is an integral component of important compound of plant cell like sugar phosphate intermediates of respiration and photosynthesis and the phospholipid that make up plant membrane. It is also a component of nucleotide used in plant energy metabolism like ATP and in DNA and RNA. The symptom of phosphorus deficiency includes stunted growth in younger plants and dark green coloration of leaves which may be malformed and contain small spots of dead tissues called necrotic spots. The leaves become dark greenish purple. Additional symptoms of phosphorus deficiency include production of slender but not woody stems and the death of older leaves. The next one is silicon. Only members of family Equisetaceae called scouring rushes because at one time their ash rich in gritty silica was used to score pots require silicon to complete their life cycle. Plants deficient in silicon are more susceptible to lodging and fungal infection. Silicon is deposited primarily in the endoplasmic reticulum, cell wall and intercellular spaces as hydrated amorphous silica SiO2.NH2O. The next one is boron. Although precise function of boron in plant metabolism is unclear, but also it helps in cell elongation, nucleic acid synthesis, hormone responses and membrane function. Boron deficient plant shows aging problem. Symptom is black necrosis of the young leaves and terminal buds. Necrosis of young leaves occur primarily at the base of leaf blade. Stem may be unusually stiff and brittle. Apical dominance may also be lost, causing the plant to become highly branched. However, the terminal apices of the branches soon become necrotic because of inhibition of cell division. Structures like fruit, fleshy roots, and tubers may exhibit necrosis or abnormalities related to the breakdown of internal tissues. The third group is deficiencies in mineral nutrients that remain in ionic form. This third group includes potassium, calcium, magnesium, chlorine, manganese, and sodium. So we will start with potassium. Potassium present within plants as the cation K plus plays an important role in regulation of the osmotic potential of plant cells. Its deficiency is mottled or marginal chlorosis, when which then develops into necrosis primarily at the leaf tips, at the margins and between veins. Because potassium can be mobilized to the younger leaves, these symptoms appear initially on the more mature leaves towards the base of the plant. The leaves may also curl and crinkle. Potassium deficient corn, the roots may have an increased susceptibility to root rotting fungi present in the soil, an increased tendency for the plant to be easily bent to the ground called lodging. The next one is calcium. Calcium ion is very common and used in the synthesis of new cell wall, particularly middle lamella that separates newly divided cells. Calcium is used in the mitotic spindle during cell division. It is required for the normal functioning of plant membrane and has been implicated as a second messenger. In its function as a second messenger, calcium may bind to calmodulin, a protein found in the cytosol of plant cells. The calmodulin calcium complex regulates many metabolic processes, which we will discuss further. So, the symptom of calcium deficiency includes necrosis of young meristematic regions like tips of roots or young leaves, where cell division and wall formation are most rapid. Necrosis in slow growing plants may be preceded by a general chlorosis and downward hooking of young leaves. The root system of calcium deficient plant may appear brownish, short, and highly branched. Severe stunting may result if the meristematic region of the plant die prematurely. So, the next one is magnesium. In plant cells, magnesium ions have a specific role in the activation of enzymes involved in respiration, photosynthesis, and the synthesis of DNA and RNA. Magnesium is also part of ring structure of chlorophyll molecule. It is also very important because the molecule chlorophyll plays important role in plant life and composed with the help of magnesium. So when deficiency of magnesium occurs, obviously it will disturb the chlorophyll formation. Magnesium deficiency is chlorosis between the leaf veins, occurring first in older leaves because mobility of this element. This pattern of chlorosis results because the chlorophyll in the vascular bundles remains unaffected for longer period than the chlorophyll in the cells between the bundle does. If the deficiency is extensive, the leaves may become yellow or white. An additional symptom of magnesium deficiency may be premature leaf. The next one is chlorine. Chlorine is also very important, found in plant as a chloride ion. It is required for water splitting reaction of photosynthesis through which oxygen is produced. In addition, chlorine may be required for cell division in both leaves and roots. Plants deficient in chlorine develops wilting of leaf tips followed by general leaf chlorosis and necrosis. The leaves may also exhibit reduced growth and bronze-like coloring. Roots of chlorine deficient plant may appear stunted and thicken near the root tip. The next one is manganese. Manganese ions Mn2 plus activate several enzymes in plant cells, especially decarboxylases and hydrogenases involved in tricarboxylic acid cycle and specifically activated by manganese. Manganese is in photosynthetic reaction through which oxygen is produced from water. The major deficiency symptom is intravenous chlorosis associated with the development of small necrotic spots. The chlorosis also occur on younger or older leaf depending on plant species and growth rate. The next one is sodium, which is very useful in C4 and CAM plant because it helps in carbon fixation. Sodium appears vital for regenerating phosphoenol pyruvate, the substrate for first carboxylation in C4 and CAM pathway. Sodium deficiency causes chlorosis and necrosis or even fail to form flowers. Sodium stimulates growth through enhanced cell expansion and it can partly substitute for potassium as an osmotically active solute. Now, we are proceeding toward group 4, deficiencies in mineral elements that are involved in redox reaction. It includes micronutrients like metal, iron, zinc, copper, nickel and molybdenum. All of these can undergo reversible oxidation and reduction reaction. They have important role in electron transport and energy transformation. They are usually found in association with larger molecules like cytochrome, chlorophyll and proteins. So first we'll start with iron. Iron has an important role as a component of enzymes involved in the transfer of electrons like cytochromes. It is reversibly oxidized from Fe2 plus to Fe3 plus during electron transfer. As in magnesium deficiency, the character symptom of iron deficiency is also intravenous chlorosis. In contrast to magnesium deficiency, these symptoms appear initially in younger leaves because iron is also immobilized. Under extreme condition, vein may also become chlorotic, causing whole leaf to turn white. Leaves become chlorotic because iron is required for synthesis of some of the chlorophyll protein complexes in chloroplast. The next one is zinc. Zinc is required as Zn2 plus for their activity and zinc also helps in chlorophyll biosynthesis. Its deficiency causes reduction in internodal growth which results as a rosette habit. These symptoms may result from loss of capacity to produce sufficient amount of oxine, IAA, indole acetic acid. Now, the next one is copper. Like iron, copper is associated with enzymes involved in redox reaction being reversibly oxidized from Cu plus to Cu2 plus. Enzyme is plastocyanin which is involved in electron transfer during the light reactions of photosynthesis. The initial symptom of copper deficiency is the production of dark green leaves which may contain necrotic spot. The leaves may also be twisted or malformed. Next one is nickel. Urease is the only known nickel containing enzyme in higher plants. Nitrogen fixing microbes also requires nickel for enzymes that reprocesses some of the hydrogen gas generated during fixation. Nickel deficient plants accumulate urea in their leaves and consequently show leaf tip necrosis. The last one is molybdenum, which is very important mineral for plant. It is present as MO4 plus to MO6 plus. It is the component of several enzymes including nitrate reductase and nitrogenase. Nitrate reductase catalyzes the reduction of nitrate to nitrite during its assimilation by the plant cell. Nitrogenase converts nitrogen gas to ammonia in nitrogen fixing microbes. The first indication of molybdenum deficiency is general chlorosis between veins and necrosis of the older leaves. In some plants like cauliflower or broccoli, the leaves may not become necrotic but instead may appear twisted or subsequently die. Flower formation may be prevented or the flowers may abscise prematurely because molybdenum is involved with both nitrate assimilation and nitrogen fixation. A molybdenum deficiency may bring about nitrogen deficiency if the nitrogen source is primarily nitrate or if plant depends on symbiotic nitrogen fixation. So here we complete this section with different types of elements which are required for the plants and their deficiency causes different types of disturbance in plant cycle. So thank you for hearing. In next section we will come with another topic, that is analysis of plant tissue reveals mineral deficiencies. Thank you.